Hello everyone, my name is Thomas. I'm from the BGI International DXBU. Today I'm really delighted to present you the BGI whole exome sequencing in prenatal diagnosis. Firstly, when we talk about whole exome sequencing, it's currently the most widely applied molecular technology for rare disease diagnosis. For rare disease diagnosis, currently more than 7,000 of the disease have been classified as rare. And taking about 300 million people population, and 50% is coming from children, and more than 80% of the disease caused by the genetic mutation. And when we talk about the molecular diagnosis method, it has been an evolution from a single genes mutation detection towards a several genes detection, towards a disease-based multiple genes detection, towards the currently most applied whole exome sequencing as an overall comprehensive evaluation of the people's genetic profile. And also whole genome sequencing in these 2 years have been widely applied in the clinical, especially for the fetus diagnosis and also the neonatal severe children's cases diagnosis. So the purpose of genetic detection is trying to find the genetic cause of the patient phenotype and its syndrome. As for the prenatal diagnosis by clinical whole exome sequencing, in 2020, there has been one publication by the ACMG to guide the use of fetus whole exome sequencing in prenatal diagnosis. It's given the suggestion majorly on these 5 parts for the fetus exome detection, suggested as the features demonstrate abnormal ultrasound image, and also after the detection of karyotyping and CMA is abnormal, is highly recommended for the whole exome sequencing as a monogenic disease diagnosis. Also, it's recommended trial testing as oral evaluation with the parent's genetic profile. Also recommended shorter TAT because it's in the fetus testing. Is better to precise its detection and given the confirmation as far as possible for the further intervention selection. Also classify the reported phenotypic, likely phenotypic, and also the phenotype, also including the reported variant classification, including the pathogenic, likely pathogenic, and also the phenotype related VUS variant. Also, given the guidance for the genetic counseling, it's required that before the testing and also after the whole exome sequencing detection. Also, for this guidance, given the other consideration, including the before testing consideration, how we're going to demonstrate the report, and also the after testing consultation, and also focus on that, emphasize that the negative report does not mean the fetus is normal. And also, the mutation pathogenicity may change. And also, the other administrative issues. For the whole exome sequencing applied for the prenatal diagnosis, there are 2 famous publications coming from, one from UK and the other one from US. For the UK detection, it calls the page detection by detecting the structural abnormalities in the fetus as a cohort study, and the other one also for the fetus structural abnormality detection, a prospective cohort study conducted in the US. For these 2 studies, all included the fetus samples around 200 to 600, and also doing the trial testing, and given the final diagnosis rate, that's with 8 to 10%. Also, the skeleton abnormalities and also the multiple malformation are the major phenotype demonstrated identified from this prenatal diagnosis. So after that, more publications have been reported for the prenatal diagnosis. So we can see from this figure that demonstrates all the reported publication that for each study ranging from 10 to around several hundreds of the sample included, And given the positive rates, the diagnosis yield from 15 to 60%, and also the autosomal dominant disease take the larger proportion regarding of the cost of the hereditary models. So the overall samples precise from these different studies that's including 3,500 samples included, and have a conclusive diagnosis yield around 70%. So that's the background that. And as for the BGI rare disease diagnosis product line, we provided target panel to detect the monogenic disease based on the customization provided by the doctor. That we provided a customized panel if the doctor selected specific gene to be interested for the detection, for the interpretation, for the reporting, and we can provide the list for them to consideration. So we provided multiple panel from 1 to 9 genes, and another one from 10 to 200 genes. Also did other specific mutation for different different disease. We also provided such service. Also, clinical whole exome sequencing is the major product for rare disease diagnosis currently in the market. In BGI also, we provide the clinical whole exome sequencing with manual interpretation, and also the exome express testing for the automatic variant interpretation. And also, we provide the whole genome sequencing for the program and for the trials as the most comprehensive evaluation of the patient phenotype. And as for the BGI experience in rare disease diagnosis and also in exome sequencing, we have processed more than 300,000 exome samples, and for each year we process more than 20,000 clinical whole exome sequencing samples for the rare disease diagnosis. Also, we have accumulated more than 40,000 clinical rare disease samples. And also, as for the academic part, we have published the paper for the discovery of the genes. More than 80 genes is discovered by BGI from the service and lab solution. So the tech transfer to localize the testing capacity in the local, including the experiment and also the analysis. Also, our testing spectrum is much more flexible. We can provide whole exome sequencing with manual interpretation, and also the express testing with automatic fast analysis pipeline. Also, we provide efficient and valid analysis pipeline with efficient variant filtering by the BGI integrated databases. And also, we provided interpretation with high clinical performance with considerably high diagnosis yield. So this figure demonstrates the overall BGI workflow from the data generation, genetic data production in the experiment part, from the DNA extraction, library construction, and capture, and also the NGS sequencing to generate the result. So the bioinformatic analysis from the alignment, variant coding, annotation, and to get the variant panning for the interpretation. As for the interpretation, we have two pipelines. One is the manual interpretation by combining the phenotype of the patient to manually interpret such variants versus the patient phenotype, given the pathogenic variant result. And if such sequencing performance is not good for this specific variant, we also do the validation to generate the final clinical report. And for the other pipeline, as for the automatic interpretation, we have provided BGI cohesion algorithm to doing the variant prioritization, and also provided variant prioritization lists for the clinical doctor for the further confirmation of the variant pathogenicity and given the final diagnosis. This one demonstrates the key features of BGI product, whole exome sequencing product. All our variants will be evaluated based on the ACMG guidance, and the mutation database is updated routinely on a semi-annual basis. Also, we reported incidental findings variants based on this ACMG guidance. And as for the testing specification, that we including all the exome regions, genes, and also covers the upstairs and downstairs 20 base pairs into the regions to evaluate any splicing effect. And as for coverage, probably high performance coverage for exon regions, the mean coverage depth of the exon is over 150x, and over 98.5% of the target regions are covered more than 20 multiples. As for variant testing tabs that we include in our reporting, including SNV indel for sure, and also exon CMV for more than 2 consecutive exon CMV that we can detect and report. And also the large CMV for all the variants more than 1 million CMV that within the target region that we can also provide such interpretation and report it in our report. Also for the other CMV-based variation like aneuploidies, triploidies, also the other mutation like mitochondrial region, intern pathogenic region, because when we design for BGI, we designed this capture region also including the intern regions to capture this clinical significance. So we will include such if there are any abnormality in the variants, including the region, intern region that we capture will be reported. Also for the other variant types, like more than 5 million of ROH, dynamic mutation will be included. And as for variant calling accuracy, that as for SNV of sensitivity more than 80% and the indel sensitivity more than 85% from our previous data. And as for BGI, previous experience, that our overall clinical whole exome sequencing positivity reporting rate for the clinical diagnosis yield is around 40%, considerably very high in the market. And this one demonstrates the prenatal diagnosis experience of BGI. We demonstrate one case, one group study that we included for the prenatal diagnosis of all the abnormal ultrasound cases in 2020. And when you take a look, around 483 prenatal samples is included in this study, and around 155 positive cases we identify, demonstrating a diagnostic yield of 32%. Also, we identify such the other specific cases like CMV cause variant around 33 cases. Also be reported as positive cases, variant, and 3 are related to the ROH mutation. Also, this figure demonstrates percentage of different abnormal ultrasound phenotype we identify, especially the multiple malformation and skeleton abnormality are the major structural abnormality that we identify from the ultrasound cases. And so next, I'm going to demonstrate the specific BGI clinical whole exome sequencing report. For this one is what we include in our report for clinical whole exome sequencing. Besides the basic information of the patient and his phenotype, we including the primary findings with the variant that demonstrate the highly related to patient's phenotype. Also be confirmed by the previous publication or identified as the pathogenic or likely pathogenic variant based on the ACMG guidance. And also we provided such evidence information like the publication and also the evidence classification. Also, as a secondary findings, we provided the variants with unknown significances related to the patient's phenotype. Also be reported in this secondary finding. Also, the other incident findings we also reported in our clinical report. Next, I'm going to demonstrate the exome splice testing. So this one compared with the traditional testing analysis pathway require manual interpretation, also with complicated pipeline, and also take time to arrange the manual interpretation. Exome splice testing utilize automatic pipeline with highly sophisticated algorithm, and we consider different aspects in this overall analysis pipeline. Also, it's extremely fast because it do not utilize manual interpretation. And the target clients of this testing is suitable for the hospital or the laboratory which have their genetic analysis capability and require high time efficiency to deliver the report, especially quite suitable for such prenatal diagnostic cases. As for this testing algorithm, is derived by the BGI, based on the BGI clinical databases. And the general workflow is we have the individual horizontal sequencing data and also the HPU series number represented by the patient phenotype. Go through this cohesion algorithm to doing the variant prioritization by the causative score. And also for this algorithm, we consider different aspects from the gene, disease databases, mutation databases, and also the in silico prediction for animal acids changes, also the other advanced analysis algorithm considering cross-species phenotype, protein interaction network, phenotypic similarity, scores, also and others will be included and combined for this variant prioritization. So this slide demonstrates such algorithm performance. We took the top 20 variants that we prioritize by this algorithm. We call it PSEA, the PSEA algorithm. When we take a look at the performance of this algorithm, the variants prioritized by this algorithm, the top 20 variants list, that's all these variants have been manually interpreted before. And the percentage of this variant identified by this algorithm, the top 20, the percentage takes account for 96%. And also when you take a look at the trial testing, it's reached up to 99% compared with the other algorithm, popular utilized algorithm for variant prioritization like SMATHER, XRARE. The performance is much higher, this percentage is much higher. And also we have published such data in the bioinformatics journal. Also for the other function is included, including the not only limit to the SMA and indel variant, but also we included the CMV and large CMV variant. Also we provide the tools for the HPU series number extraction based on the patient, the doctor description of the patient. Also exclusion the other partial cytogenes inference. And to be more specific at what's included as the workflow, firstly we need to have the genotyping data, the horizontal sequencing data generated by the sequencer, also the phenotype data represented by the HPU series number. Go through this cohesion algorithm to do the variant prioritization, and will provide the variant prioritization list according to the probability of the pathogenicity, and further given to the doctor for manual interpretation and to generate the clinical report. And this one demonstrates the report template that not only limit to the basic information, we provided the testing findings with the ranking of the variant prioritization prioritized by this algorithm, including the genes information, variant coordinates, and also is related autoimmune disease, and also the heredity models. And also, we also reported the incident finding if such variant is identified. And not only limited to the top 20 variant list, but also the other variants prioritized by this algorithm are all included in one Excel data. Also in this Excel that we contain all these prioritized variants regarding its information in different databases, and also its ranking by this algorithm, and also the sequencing index. So next, we're going to demonstrate some selected demo cases. First one is this, we talk about phenotypic heterogeneity. We identify with the same genes with a different mutation that demonstrate the different phenotype in the fetus. We take a look at this case with COL2A1 genes with type 2 collagen related disease. For these genes, its coding protein is mainly distributed in human cartilage, visual body of eyes, inner ear, extracellular space of calcium binding protein, and extracellular matrix. The mutation of this gene results in abnormal protein function that can lead to a range of different dominant genetic disorders, such as type 2 collagen-related disease. And when we talk of the specific mutation type, like the missense mutation in the dominant model, that the glycines that is replaced by the other amino acids, that we actually, from our previous experience, we observe very serious clinical phenotype in the fetus, resulting the tolerance type of flattering vertebral fetal skeleton dysplasia. Like the cases we identified from these 5 fetal cases, very severe clinical manifestation was observed, like the repeated kidney and also the malformation, overall malformation. Also the long bones was observed with short, and also the dilated dwarfs, short limb bone deformation, all is observed, especially at the fetal status. But when you take like the other kind of mutation, like the meaningless frame-shaped mutation, leading to the premature termination of the translation of such protein, such mutation affecting the collagen synthesis, but with the clinical manifestation is not severe, it's relatively mild, such as the Stickler syndrome, type 1, and the other eye abnormalities. And also from our previous experience with this type of the mutation, we majorly observe such mutation in the adults from 17 to 30 years old. And we can observe the different eye disease phenotypes, like the retinitis pigmentosa, and also the punctual license in both eyes, and also the other like kinds of phenotype, like, such as cleft palate and stemmer dysplasia. But still, compared with the fetus phenotype, this is much mild. The second case I'm going to demonstrate is for this gene, FGFR3, for the fibroblast growth factor receptor family genes. And this gene records a family of fibroblast growth factor receptor. It's very conserved in the different species. It plays a major role in the bone development and maintenance. And mutation of such genes will lead to the abnormality in the skeleton. And also, from our experience, we observed that such specific gene mutation will cause the specific disease, which we call the pleiontrophy. And as for the 2 specific phenotype that we observe from the fetus, one is the achondroplasia, the other is the chondrodysplasia, with different phenotyping severity. Like for the achondroplasia, it's much severe that majorly is identified with manifestation within 1 year after the birth, and still demonstrate very severe manifestation during the ultrasound under the pregnancy of the lady, that's including the chondrodysplasia, also the protruded short statues, and also the children fingers can be observed. But as for the other phenotype, chondrodysplasia, even though the phenotype is mild, but it's really difficult to differentiate from the other cancer type. So that's we must need a genetic detection to identify such cause of such manifestation. And this one demonstrated BGI previous experience when we identify such mutation from the fetus and also from the other cases on these FGFR3 genes. Even though we can see located on the same, the mutation is same that claim the cause of this disease, but the manifestation, no matter for the fetus and also for the afterborn children, is quite different, like ranging from label long, bone short, considerable probability of short limb deformity, and also like the thin livers long bones, and it's observed. And also as for the other children phenotype like short statures, and also the dwarfism also can be observed. So still, we have been doing the genetic testing, like whole exome sequencing testing, to identify such pathogenic variant, even though the manifestation is quite difficult for the doctor to identify the specific mutation, and we have to utilize whole exome sequencing and genetic detection to give the final confirmation of such manifestation. And the third case is coming from the our exome express product that we utilize automatic variant prioritization algorithm to identify such cases. This one is related to the Noonan syndrome 8 case. Also happened on the fetus cases that we observed non-immune hydrophytus, and also the hydrocephalus we observed from the patients. Also the enlarged lungs between the eyes is also observed in the fetus, and for such cases is transferred for the microarray detection, but the result is normal. No mutation is identified, and we go through the exome sequencing testing, and we're doing the variant prioritization in our top one variant that will identify such mutation in IRT genes that from the previous function study that demonstrated this mutation could increase the binding of the IRT to the other factors that affected the serum activation compared to the wild type, and also such altered actin-dependent process, such as the cell spreading, adhesion, and migration, may explain the certain clinical features in the fit patients with such Noonan syndrome. And this one has been published by BGI by cooperating with the one Taiwan hospital. Okay, next, I'm not only limited to the service, but I'm going to have a brief introduction of the BGI lab solution. So this one demonstrates major devices and reagents that we can provide for the localized whole exome sequencing testing in our partner slide. From the DNA extraction and library construction, we can provide the SP100 device for the automatic sample handling from the DNA extraction to the library construction, and also the exome capture that can perform on this platform. And also, as for sequencing, BGI, we have our own sequencing machine that we provided the MJDL200 and also the DNB6 G400 sequencing as a major platform for the whole exome sequencing. Also, we can provide the DNB6 T7 platform for large throughput whole exome sequencing needs. And also, we can provide the BGI Hello system for the localized bioinformatic and variant interpretation, and also is served for the report generation and also handling for the sample and sequencing management as overall software to fulfill the variant analysis and report generation. And now, so that's the device part. And also, we provide the reagent, including the DNA extraction and also library construction and also the exome capture reagent, and also the sequencing reagent for the different platform G400 and T7 for the different specific. Thank you for your consideration. We hope our technology could help you to have a healthy baby, and we hope there will be less rare disease cases in the future to achieve a better life for the globe. Thank you.